Hey everyone, this is Stephanie Sippa. I'm going to talk to you a little bit today about the use of olive extracts as a possible apoptosis inducer in HL60 cancer cells. To start off, I'm going to give you guys a little bit of background before I get into the results of my study. In 1977, the HL60 cell line was established from a patient with acute myeloid leukemia. These cancer cells are trapped in the myeloid progenitor phase and keep dividing instead of differentiating into different white blood cells. My extract is based on a study in 2006, which found that extracts from olive skin can inhibit proliferation and induce apoptosis in HT29 human colon cancer cells. They also believed that the Mediterranean diet that involved a lot of eating olives and those kinds of foods were found to be protective against colon cancer. Apoptosis is programmed cell death, which occurs in steps of shrinking and blebbing, and ultimately the cell comes apart in tiny little blebs, which is considered to be non-harmful when you compare it to necrosis, which releases all the things inside the cell into the surrounding environment. So apoptosis is the goal when we're testing these olive and other extracts against these cells. In order to collect data and determine my results, we used the confocal fluorescent microscope and the viability assay. The microscope would be used for fluorescent microscopy. Fluorescence microscopy helps detect morphological change in the HL60 structure and can show if the extract is actually inducing apoptosis by giving us a visual on what state the cell is in. The viability assay shows if the extract has any effect on cell growth by measuring cell viability. And this is done by measuring the reduction of WST8, which only viable cells can do, and it means that if they're reducing it, the cells are healthy. So when I was collecting data, I was looking for lower absorption rates, and that would mean that the extract was killing off the HL60 cells. These are the viability assay results. One is done after 24 hours and one was done after 72 hours. After initially doing the 24-hour one, I considered if the olive extract was maybe a long-term apoptosis inducer and it took longer to actually kill off the cells. Although the t-test did not determine my olive extract results significant, you can see that the absorption rate did go down between the two. It makes me ponder if even longer exposure to the extract would have killed even more and made the results significant. The controls in here were these cells with the media, a 70% ethanol extract, and camptothecin, which is a positive control known to induce apoptosis. And as you can see, it was my only significant result during the viability assays. This is one of the photos of my fluorescence microscopy under the confocal fluorescent microscope. We use the HL60 dye on this one. The HL60 dye is a DNA binding dye. Because my viability assay results were so non-significant, I didn't expect to see a lot of evidence of apoptosis in the microscopy. Possibly this cell right here could be showing some condensation when you look at this photo from the Lab 5 PowerPoint. This cell down here could possibly be showing some condensation, as well as this one. Their marks seem to be a little bit brighter than the other healthy cells. I didn't notice any fragmentation or apoptotic bodies in my photos. And honestly, most of these cells are normal healthy cells because my extract did not appear to have much of an impact on these HL60 cells after 24 hours. When I compare my cells to the controls from class, my cells appear to look a lot more like the ethanol-treated cells than the camptothecin-treated cells. In these ones, you can obviously see a lot of condensation and or possibly fragmentation in this location right here. So again, this is just further showing that my extract didn't have much of an effect after 24 hours. After the 24-hour fluorescence microscopy, I decided to do a 72-hour treatment. This might have more of an impact if you look at the cells. This one could almost be fragmenting. It's unclear just because of the quality of the photo. there appears to be a little bit more condensation happening in these cells. They don't look quite as round as the healthy cells do. If you look at the shape of this one versus this one, you can obviously tell that there's some sort of a difference here, possibly some sort of effect. This again supports that maybe olive extract takes more time to create an apoptosis effect in HL60 cells. There would have to be more data to support this. The next step to determining the apoptotic properties of my olive extract was to perform a COMET assay. This was done by suspending the HL60 cells that had been treated with my extract for 72 hours in cold PBS. Then we put the slides in agarose solution and pipetted onto the COMET slide. The slide was then put through gel electrophoresis and then stained with green dye. The purpose of this is to see the DNA tails. If the cell is going through apoptosis, the DNA is going to fragment into tiny pieces. So because DNA has a negative charge, when put through gel electrophoresis, the negative pieces of little DNA are going to move towards the positive side. And if the pieces of DNA are smaller, you're going to see a longer tail. And therefore, it was further along in the apoptotic process, and your extract would have a better apoptotic use if they have the bigger COMET tails. On my slide, you can see a very small tail. I would categorize this as a big old 0. It's very small. It looks like it was just starting after 72 hours. When compared to the camptothecin slides, you can see that it has much bigger tails of DNA for where those were. I would categorize those as possibly 1s or 2s because they're longer. Again, this COMET assay just shows that the olive extract does not have much of an apoptotic effect after 24 or 72 hours. So in conclusion, my olive extract shows promise as a long-term inducer of apoptosis in HL60 cells. The viability assay showed that there was a lower viability in cells after 72 hours as compared to 24 hours, though it wasn't significant. But in the COMET assay in the fluorescence microscopy, you can see that cells were starting to go through apoptosis, and the COMET tails were just starting after 72 hours. This means that a longer time for the treatment to work on the cells could induce apoptosis in these cancer cells. So for future directions, I would try a longer time for the extract to work on the cells. I would also look at making my extract more concentrated. In the study that I base this off of, they got their extracts from just the skin of the olive. Now, I'm not skilled enough, and I couldn't get just the skin off of these olives, so my extract was mostly from the whole fruit itself. I would also try different lines of cancer cells. Maybe HL60 leukemia cells aren't what olives can attack. And they appeared to work on colon cancer cells in the study that I found, so maybe I could repeat this with those types of cells in the future. The study also talked about the Mediterranean diet, as I mentioned earlier. They said that eating olives could have benefits to help prevent cancer in the first place. So possibly I could look at long-term studies of the amount of cancer that people develop based on how much or how many olives they eat versus someone who doesn't eat any olives and their risks of cancer on that. One conclusion I could possibly make is that maybe olives don't induce apoptosis, but maybe they prevent cells from becoming cancerous in the first place. And there are a lot of studies I could do to try and figure that out. But for now, it just looks like a longer term study, and a more concentrated extract could possibly show the results I want for an apoptosis-centered study. Here are my references. I hope you all enjoyed listening and learning a little bit about the possibilities of olives as a cancer treatment and/or cancer preventer. I hope you all have a nice day, and goodbye.